Hey everyone, nice to see you. I'm Mr. VaughnHuffer. Thanks for tuning in for another instructional video. This video is about muscles and signaling between muscles and neurons. As you can see from the picture, our body is filled with muscles. They're the reason we can move and hold ourselves up against gravity. Also, muscle contractions even help circulate our lymphatic fluid around the body. But muscles are more than they appear. In this video, we are going to talk about the general skeletal muscle anatomy. We're going to talk about sarcomeres, which are the basic units of muscles. We'll discuss muscle contractions. We'll also discuss signaling between neurons and muscles that bring about muscle contractions. And lastly, we'll talk about lactic acid fermentation in muscles before discussing an AP-style question with included answer. Within a muscle, say the bicep, there exist muscle fibers called myocytes, which are further subdivided into long strings called myofibrils. And you can see this in the image up here. We have our myocytes. And then we have these long strings, myofibrils. But even these myofibrils are made up of smaller units. And these smaller units are called sarcomeres. And you can see a sarcomere right here on the picture. So the most basic functioning unit of striated muscles, these sarcomeres, or units of muscles, are connected to one another by the transverse tubule, a tube of the plasma membrane that crosses through the inside of the myocyte to ensure that the signal reaches all of the sarcomere units within the myofibrils and within the overall myocyte. Muscles also have a unique structure called the sarcoplasmic reticulum that stores and releases calcium and activates muscle contractions. So we'll discuss this more in later detail. Just to reiterate, a sarcomere is the basic contractile unit of muscles. This means that basically a sarcomere is a micromuscle. Sarcomeres are comprised of an arrangement of proteins that work together and interact with one another, and two of the most important proteins at play are G-actin, a globular protein subunit, that are linked together into an actin filament that resemble a beaded necklace. And you can see this right here, this little beaded necklace looking actin filament. It's also represented up here. Oops. Let's go ahead and erase that because that's actually not actin, that's myosin. So you see the little actin filaments right there on this sarcomere unit. The second important protein is myosin. So myosin come together like a twisted string or a rope with lots of braids and form a structure called the thick filament. And you can see that right here in the image. So this is the thick filament, and down here to the bottom is a myosin molecule. And you can see that they're braided together and they have these two little motor heads. So on this thick filament, the myosin heads act like hands, pulling the filament down the actin ladder during muscle contraction. So right here, these myosin heads pull down the actin filament, or they walk down this way, which actually causes a pulling towards the middle of both of these little Z discs here, or these little end plates of the sarcomere. So the Z discs are pulled towards the center, which causes muscle contraction. Another protein complex shown is the troponin-tropomyosin complex. So this complex provides regulation to muscle contraction by either covering or revealing the binding sites on the myosin heads, or for the myosin heads on the actin filament. And you can see those protein complexes here running on the outside of the thin actin filament. And so you have the troponin, which are the little green dots, and then you have the tropomyosin, which are the long strings that are holding them together. And these guys are just sitting here covering the binding sites on the myosin for these little, or actin on for these little myosin heads. So an important takeaway for this process, though, is that muscles, by way of myosin and actin, convert chemical energy from ATP to physical energy of motion. So while it may seem relatively complicated at first, the process by which muscles contract is pretty straightforward and also cyclical. So this means that it is a process of repeated steps that happen over and over. So again, within a sarcomere, the basic contractile unit of muscle, the thin filaments made of actin are anchored to the outsides, while the thick filaments made of myosin are sandwiched on each end by the actin filament. And you can see that in the little GIF I've provided in the bottom right. So you have your myosin heads, and then you have your sliding actin filaments. An important thing to remember is that this process does not happen with all of the myosin heads instantaneously. So imagine 10,000 people at a marathon starting line. The gun goes off, and not all the people on the starting line are going to start at the exact same time, nor do all of the people start at the exact same speed. It is going to be a wave of action until all people have started the race and are running. So imagine that we are a single myosin. So myosin heads, first of all, cannot bind, or without a bound ATP, cannot bind actin. And so the first thing that is going to happen is that the myosin starts detached from an actin with a bound ATP molecule. So once the myosin hydrolyzes the ATP to ADP, it changes shape. So this shape change is called a conformational change. And now it's able to bind the actin filament. So next, once the myosin binds to the actin filament, this causes the myosin plus ADP to pull the actin filament towards the center of the sarcomere unit. So then, the completion of this pull, which is called a stroke, like the stroke of a paddle in a canoe, causes the ADP to dissociate or fall off of the myosin head. Then the empty binding site left by ADP allows a new molecule of ATP to bind to the myosin head, which then triggers the myosin head to dissociate or let go of the actin filament. So after this cycle has completed, the myosin head is now able to complete the cycle again. And you can, again, see an animation of this in the bottom right. So next we're going to talk about signaling of muscle contractions. The contraction of sarcomeres and muscles is signaled to the muscles by associated neurons. Neurons interact with the muscle at the motor end plate located at the neuromuscular junction, so the part of the muscle where neurons interact with the muscle itself. The muscle side of this junction is packed full of neurotransmitter-gated ion channels that only open when they bind the molecule acetylcholine, which is a neurotransmitter. So if you remember learning about membranes, then you should recall that membranes are electrically polarized, with the inside of the cell having a negative charge and the outside of the cell having a positive charge. Furthermore, the concentration of sodium ions outside of the cell is much larger than the concentration of sodium ions inside of the cell. So when the gated ion channels at the motor end plate bind the neurotransmitter acetylcholine, this causes them to open and allow positively charged sodium ions to flood into the cell towards the internal negative charge and down their concentration gradient. This flood of positive ions causes a depolarization of the plasma membrane, or an internal change from negatively charged to positively charged due to all of the sodium ions that have flooded into the cell. This, in turn, causes voltage-gated sodium channels further away from the motor end plate to open due to the change in charge on the membrane. And this depolarization continues to spread outward like a wave. This process is called an action potential and is demonstrated by the GIF at the top right. So in this GIF, it is showing an action potential that is started, and then these voltage-gated channels open and allow ions to flow through. And if you notice this line here at the bottom, right, it actually shows you the change in polarity, so along with the outside and the inside. So this action potential carries the signal started by the neuron to the sarcoplasmic reticulum inside of the muscle cells by way of the transverse tubule, or the T-tubule. The sarcoplasmic reticulum, again, is an organelle like the ER, except that its purpose is to store and release calcium ions into the muscle cell. Calcium ions are used as the intracellular, or inside the cell, signal for muscle contraction, and because of this, intracellular concentrations of calcium are kept really low. An important thing to know is that ATP is always present at high concentrations inside of muscle cells. Also, myosin heads are always ready and waiting to go into action. However, the binding sites for myosin on the actin filament are physically blocked by the troponin-tropomyosin protein complex. So this protein complex is shown here, and the change of covered to uncovered is also demonstrated. So when the sarcoplasmic reticulum receives the action potential, it releases a flood of calcium ions into the cell. These ions bind to troponin on the troponin-tropomyosin complex and cause it to undergo a conformational change where it slides off the myosin binding site of the actin molecule. This allows the myosin to jump into action where they start binding and undergoing a series of conformational changes shown previously to bring about muscle contraction. Because sodium and calcium are so important in muscle contraction, this is why sports drinks always boast about how many electrolytes they have or how perfect and how optimized the electrolyte balance in their drink is. Electrolyte is a fancy term for ion like sodium or calcium. So if your body does not have enough electrolytes or the correct balance, it cannot as effectively signal your muscles, and you either become sluggish or start developing muscle cramps. So the images provided just give you a little bit more clarification of how this whole signaling and contraction of muscles works, from the action potential and a demonstration of that. We also have another demonstration of the action potential and signaling at the neuromuscular junction in this GIF right here in the center. We have the troponin, tropomyosin, and how it interacts with the actin molecule down here at the bottom. And then up here at the left, you have the overall schematic of the signaling and where these things are located in terms of the neuromuscular junction right here, and also the sarcoplasmic reticulum, T-tubules, and the sarcomere. So sometimes our muscles require more oxygen in a situation than we can physically provide them. Imagine you're running full speed from danger to save your life. Your muscles are cranking out ATP, but you're using so much that your body can't circulate enough oxygen in this unusual circumstance to meet the demand. But your muscles still need ATP. Evolution has the solution. Instead of aerobic respiration, your muscles begin to perform anaerobic fermentation, also called lactic acid fermentation. So when we exercise intensely, our muscles begin to use more oxygen, producing ATP in the electron transport chain than we can physically supply. This causes a near shutdown of the electron transport chain and the production of ATP, because the electron transport chain requires oxygen as the final electron acceptor. Furthermore, the byproduct of respiration and the production of ATP is the regeneration of NAD+ from NADH, a molecule that's used in the electron transport chain. So NAD+ is a required molecule in glycolysis, a process that does not require oxygen, and also is required for cells in order to survive. So without adequate NAD+, cells cannot perform glycolysis. So to remedy this shortage of NAD+, the cell begins to divert pyruvate molecules from the electron transport chain and instead use those pyruvate molecules to convert NADH into NAD+. This ensures that the cell can still produce some ATP through glycolysis, even in the absence of oxygen. The downfall is that the cell instead produces lactic acid that begins to build up in the muscles and causes a burning sensation and potentially can cause muscle cramping. Occasionally, this buildup lingers and is oftentimes the cause for sore muscles the next day or two after working out. But you can help remedy this by stretching, massaging, and soaking your sore muscles to aid the body in the removal and breakup of the lactic acid. So in this section of the video, we will answer a question relating to the topic of muscles, muscle contractions, and signaling. And this question is taken directly from College Board. So the question says, explain how membranes participate in the following biological process. And the process was muscle contraction. So a couple of things that we need to mention when answering this question. There are two places where membranes, or two very critical places where membranes play a role. The first one is in the signaling at the neuromuscular junction. All right, so this is done through membrane signaling. And then the second very important part is the membrane system that is the sarcoplasmic reticulum. So this membrane system stores and releases calcium into the sarcomere to signal muscle contraction. And so some vocabulary to use I put over here: sarcoplasmic reticulum, of course that's what releases the calcium. The motor end plate, so this is our first site where membrane signaling happens. Then we have acetylcholine. This is the neurotransmitter that is sent across the neuromuscular junction. Then we have troponin, which binds the calcium released by the sarcoplasmic reticulum. So don't forget troponin right here. And then we have depolarization, which is what happens to the membrane of the neuron or of the muscle in order to signal to the sarcoplasmic reticulum. And so to answer this question, at the start of signaling for muscle contraction, the neuron associated with the motor end plate releases acetylcholine that bind to the acetylcholine-gated sodium channels on the motor end plate membrane and cause them to open. This flood of sodium into the cell causes an action potential, depolarization of the membrane, that acts to send the signal further down the membrane to voltage-gated sodium channels that propagate the signal to the sarcoplasmic reticulum. Once the action potential reaches the sarcoplasmic reticulum, the change in voltage triggers the sarcoplasmic reticulum to release calcium ions into the muscle cell. The calcium binds to troponin, covering the actin filaments, and causes a conformational change that reveals myosin binding sites and allows myosin heads to bring about muscle contraction. So in our answer here, we have talked about this signaling across the membrane by acetylcholine, all right, and how it binds to channels on the motor end plate membrane, right? And then we've also talked about action potential and depolarization of the membrane, because this is key. The signal would not reach the sarcoplasmic reticulum if we did not have this action potential going down across the membrane into the T-tubule and reaching the sarcoplasmic reticulum. And then we've also mentioned about how the sarcoplasmic reticulum as a membrane system releases calcium into the muscle cell, and how this calcium then affects a change by binding to troponin and ultimately revealing myosin binding sites that allow them to bind and bring about muscle contraction. Thanks again for tuning in, everyone. I hope you found the video useful. And if you have any questions or need any help, feel free to contact me in Google Classroom. You can always contact me by email, hit me up in our Google Chats Hangout, or you can sign up and video conference with me live. Hope everyone has a great day, stay safe, and stay home.